So I'm thrilled that we have Carey Hannaford joining us today. Carey is the director of MGPalaeo, which are geological consultants specialising in palynology, mainly servicing the oil and gas industry for more than 20 years. She'll be chatting with us today about palynology in exploration and the importance of having a good stratigraphic database. So she'll be going through what it is, how it's used, and how we're now using it in mineral exploration. So it's going to be a great session. I hope you all enjoy it. Please use the chat. You'll have the chance to jump off mute and we'll open up the floor at the end. And yes, thank you so much, Carey, for joining. It's just wonderful having you.
Hi, thank you. Now I'm new to all this technology and I have just found out I have a bus stop right outside the office, so I'm hoping that the sound comes through okay for everyone. Good. So yeah, I'm happy to be here today, and after that great start, I'm going to put you all to sleep, I'm sure. I'm a palynologist and most people find us incredibly boring. So I'm going to try and make it less boring for you, but we'll see how we go. So I want to, yeah, as mentioned, talk about the importance of having a good stratigraphic database, and to do that I'll have to talk first about palynology, which is my favourite subject, and probably we'll see if everyone's bored, but some of those jokes earlier on kind of tie into it, because there's a bit of pollen and a bit of bees in here, and a little tiny bit, and some bugs, but a different sort of bugs to these spiders. So before I start, as a bit of a background on what we do, we're a geological consultancy company which has core operations around biostratigraphy, and our main focus is palynology. So although the bulk of our work is centred around the oil and gas industry, we also do a lot of work for coal exploration, coal seam gas, a lot of work for the universities in different fields of research, as well as sedimentary mineral exploration. Most recently, that would be uranium exploration in South Australia. We also work in carbon capture and storage, and a huge amount of our work recently has been in hydrogeology as well. So we also do rig site biostratigraphy. So this is basically looking at these bugs on the rig, and we're really not very popular out there because we're very weird. And most of that work these days is being done in the highlands of PNG. So not the greatest job with 6 weeks on, 2 weeks off usually. So it's not a lot of fun from the PNG highlands. In recent years, we've also diversified out to building our own, starting with our own in-house stratigraphic databases, and then that sort of evolved into building stratigraphic databases for other people. So what we found when we were constructing these databases was that it was really important to have a standardised and detailed stratigraphic framework. So that's what I'm going to try and talk to you today, because that's how the biostratigraphy translates across to a stratigraphic database. So my talk today will have two main parts: a background on biostratigraphy. This is what forms the core of most of what we do. And the second part on the importance of a good stratigraphic database, with some insights into why they're useful and how to build one. I will use some examples from the Otway Basin and the Great Artesian Basin, where we've built some large databases which are available for free from various government websites. I'll put those links up there as well. What is biostratigraphy? Very briefly, biostratigraphy is the branch of stratigraphy that deals with the distribution of fossils in the stratigraphic record. It's the use of these fossils to establish relative ages of rock and to correlate sedimentary successions within and between depositional basins. Through knowledge of key fossil species that existed for short periods of geological time, we can use the occurrence of these key species to interpret the age of strata in the sedimentary basins in which we explore. The second term you'll hear today is the biozone. This is an interval of strata characterised by a range or abundance of certain fossil taxa. We can use evolutionary points, extinction points, acme points, which also have a paleoenvironmental significance. For example, flooding events, climate events, and mass extinction events, all of which I'll discuss in more detail through the course of the presentation. So there's a bit of jargon related to this field, but I'll try and keep it to a minimum. Biostratigraphy centres around a branch of paleontology related to microfossils, which are described as the microscopic remains of an organism or part of an organism which has been preserved in the geological record. There are three main groups of microfossil. We have either microfossils themselves, which are typically calcareous or siliceous, nanofossils, which are nanoscale microfossils, and palynomorphs, which are the organic walled microfossils. I'll expand on these a little bit later, but most of what I'll be talking about today will be the palynomorph category, as this is what forms the basis of the Mesozoic and Cenozoic stratigraphy in Australia. So depending on which part of the section you're working with, there will likely be a different microfossil group best for the job. And this slide here should just show the black, thick black lines there are sort of where each of these fossil groups are most useful in the stratigraphy. So if you're working with Cenozoic carbonates, for example, then you will likely be targeting foraminifera and nanofossils. In the early Cretaceous, you will have a choice of palynology or microfossils and nanofossils. Thus, you would be then considering if you're drilling in a predominantly siliciclastic environment or a carbonate-dominated succession. Through the Triassic and late Paleozoic, it is likely that palynology will be your main biostratigraphic tool. And if chasing older Paleozoic targets, then you might consider conodonts or acritarchs. So basic vascular plants first evolved in the Ordovician, and thus spores have been present in the fossil record from about that point. However, spores are only really become abundant enough to be used for stratigraphic tools from the Devonian onwards, and that's closely followed by gymnosperm pollen from the late Paleozoic onwards. Along with spores and pollen, the most widely used palynomorphs in industry biostratigraphy are actually dinoflagellates, otherwise known as dinocysts. These are first evolved fossilizable cysts in the mid-Triassic, but they're particularly useful marker fossils in the late Jurassic and Cretaceous, and they give us a much higher resolution than the spore pollen, so they're quite useful, but you need marine sections, unfortunately. So each of these key palynomorph groups has different ranges and so different stratigraphic utility. So I'm going to start off with palynology because it's my favourite subject. So why do we use it? Well, microfossils are abundant and well preserved, and you get thousands of little fossils from as little as about 10 grams of sample. So they also have a high evolution and extinction rate and can give us very high resolution. And the marine microfossils are globally widespread, which makes them useful correlative markers across the globe. Palynology is particularly useful to us in the late Paleozoic and Mesozoic siliciclastic successions, and it represents by far the most widely used fossil group in Australian hydrocarbon exploration. It's also very widely used in similar siliciclastic dominated successions in the North Sea, West Africa, parts of the Middle East and North Africa. It's less widely used in Southeast Asian and Gulf of Mexico areas where foraminifera and nanofossils are typically the key fossil groups. In the next few slides, I'll just give you a very quick summary of the main fossil groups that we can use for biostratigraphy. So there are 6 main palynomorph groups: spores and pollen from terrestrial plants, single-celled dinoflagellate cysts, and acritarchs that mostly relate to aquatic settings. So I'll go on to this next slide here with spores. Spores are produced as part of the reproductive cycle of lower vascular plants, such as lycopods, horsetails, and ferns, along with some algae, fungi, and protozoa. As these spores are a unit of sexual or asexual reproduction, they are often adapted for dispersal and survival, often for extended periods of time in unfavourable conditions. For this reason, they've developed very robust organic walls. They are also chemically inert. So both this robust physical framework and the chemical stability mean that the spores are extremely well preserved in the fossil record. Once they're buried in the sediment, away from oxidising surface environments, there is a high likelihood they will be preserved. Unlike other fossil groups that are preserved as imprints or recrystallised forms, palynomorphs remain as their original organic structures. Spores are important biostratigraphically in the late Paleozoic and Mesozoic, where they are often the sole useful biostratigraphic tool in non-marine environments. However, whilst they're derived from terrestrial environments, they can also be washed into the marine sediments by fluvial systems, so they're also useful in marine systems. Up the top of the screen there, we have some examples of spores. On the far left, you've got some Devonian, a very important Devonian spore. And then next to that, we've got a Permian one. That's one of my favourite Permian spores, very used for the late Permian. The one next to that is a Jurassic marker, and one that has caused me great grief in my career, so I'm not a big fan of that one. And then on the right-hand side, all 3 of those spores are Cretaceous spores. So next we have pollen, of which there are 2 types. Gymnosperm pollen is produced by seed-producing plants such as conifers, ginkgos, and cycads from the Carboniferous onwards. They produce naked seeds, that's seeds that are not protected by a fruit, and extant gymnosperms are mostly woody trees and shrubs. Many gymnosperms produce a pollen with two air sacs attached to a central body. And you can see some examples in the bottom left corner there of some Permian bisaccate pollen. These are specifically designed to be transported long distances by the wind. Such gymnosperm pollen is particularly useful for biostratigraphy in terrestrial deposits in the Carboniferous, Permian and Triassic. So if you've ever been near a pine forest at the right time, you will have found, perhaps if you've driven your car through there, a yellow dust everywhere. That yellow dust is pollen. And if you were to look at it under a microscope, you would see something like this. And this here is a scanning electron image, but you have a central pollen body, and then those two things below are the two air sacs, which allow it to fly around. And these things have remained largely unchanged since the really early ones, which, well, examples of which you can see those Permian ones sitting down there. Angiosperm pollen is produced by the flowering plants, and they're the most diverse extant plant group. With around 300,000 species, they represent approximately 80% of all known green plants now living and have an enormous variety of body types and forms, ranging from annual herbs to climbing vines to massive trees. In contrast to this, there are only roughly about 1,000 described gymnosperms today. So angiosperms first appeared in the early Cretaceous, becoming common elements of the late Cretaceous pallinifloras, but only replaced conifers as the dominant tree types in the Cenozoic. So they represent important elements biostratigraphically from the late Cretaceous onwards. And there's bottom right-hand corner there is a few examples of some Australian late Cretaceous angiosperm pollen. Okay, so I just want to make a little aside here because I love palynology and here's some cool stuff. So on this next slide, I have an example of some modern pollen work that we've done in the past. And this came from a very interesting research project on some beehives. So up in that top left corner there, we have a kind of a core of pollen, which came from a working orchard beehive. And here you can see there's a number of different layers, which you've got letters next to there. Each one of those layers had a different dominant pollen. Layer H, there, that little sort of off-white coloured layer. This one was almost 100% wattle pollen, which has a really distinctive morphology. And as a tie back to what I'm originally talking about, the fossil record, if you click one more time, we should get a picture of a Acacia pollinites myriosporites, which has an evolution point in the early Miocene and is one of our main markers for the spore pollen in the Cenozoic in Australia. So we get a really, really interesting range of morphologies here, and we can tie some of this Cenozoic stuff back to the modern pollen. So this is my favourite group. This is the area I specialised in my early days, Dinoflagellates. So these are perhaps best known by the general public for the red tides that they form. And they form a major base component of the marine food chain. Dinoflagellates are a unicellular motile algae characterised by a flagellum that encircles the body and also one that in the transverse direction, which allows it to kind of spiral through the water column. They're primarily autotrophic, which means they photosynthesize, and so are most common in the photic zone in the ocean. But there are also heterotrophic, which are food-eating ones, and photosynthetic symbionts also found. So dinoflagellates have a two-stage life cycle where they have a floating stage where reproduction occurs, and eventually the organism will shed its flagella and a cyst will form around it, which will sink through the water column to the seabed, where it will rest until a time when the water conditions become optimal again, at which point the cell will exit the resting cyst and the cycle starts all over again. And it is this resting cyst which is preserved in the fossil record. So while we're on the dinoflagellate one there, you can see there's some dinoflagellate cysts in the bottom right-hand corner there. And you can see at the top of it, they've all got this little sort of escape hatch where the cell escapes from the cyst. So we'll go back to the acrotarchs, which was the next slide. This is basically a dustbin group for a number of different algal types, a few of which we know the origins of. For example, the resting cysts of the chlorophytes, or green algae, are included here. And then the top left-hand corner there, acrotarchs. So they likely represent an ancestral group to the dinoflagellates, and they are in the rock record from the Precambrian to recent, but they have their highest biostratigraphic utility in the Paleozoic. On the right, we have some algal groups, Prasnophyte algae, which is in the rock record from the late Precambrian to recent. and they are the planktic cysts of green algae. The value there is environmental and source rock rather than biostratigraphic, but we still count these for interest's sake. So then we also have the freshwater algae. For example, Botryococcus, which ranges from the Ordovician to recent, and Pediastrum, which appears in the Cretaceous but is not commonly found until the Paleogene. So both forms are colonial algae and form dense blooms with high oil source rock potential. And the last two groups are Scelechodonts, which are the jaws of some marine worms. Fossil Scelechodonts are particularly common in the Ordovician, Silurian and Devonian marine deposits, but the organisms they come from have existed in the rock, on the planet from the Cambrian to the present. And the vast majority of these are marine species. So, and Chitinazoans on the right-hand side there, we're not really sure what they are. It's been suggested that they have a protist plant or fungal affinities, or that they may represent eggs of juvenile stage of a marine animal. However, most recent research suggests that they represent the hard shell of a group of protists with uncertain affinities. The ecology of Chitinazawa is also open to speculation. Some may have floated in the water column, while others may have attached themselves to other organisms. Most species are very particular about their living conditions and tend to be most common in specific paleo environments. So that's what we use them most for. And they live in the rock record from the Cambrian to Carboniferous, but they're most common in the Ordovician to Devonian. So that's the organic fossil group. So I'm just going to summarise in the next slide the microfossil group. And these are everything but the organic stuff, basically. So we have foraminifera, which are single-celled protists that produce a chambered shell, and they may be either planktonic or benthic. So the benthic ones are very useful for paleo bathymetry studies. Although they range in age from the Cambrian to recent, they're principally used as a biostratigraphic tool by the oil and gas industry from the late Cretaceous to the Cenozoic. We have Austropods here, or sometimes known as seed shrimps, and these range from the Cambrian to recent. They need a lot more sediment, so we don't use them as often in biostratigraphy because we need bigger sample sizes. So the next one is the Diatoms, which are large single-celled creatures that live in a benthic or planktic habitat. Again, they have a siliceous skeleton, so we don't see them in our palynological preparations because the process of extracting the fossils from the rock means we have to dissolve the siliceous materials, so these guys get dissolved. They're common in the rock record from the late Cretaceous to recent. Radiolaria are really, really pretty, but again, they have these are planktic protists with skeletons composed of opaline silica. So their ranges from the Ordovician to recent, but they're most common since the Jurassic. And they may occur in abundance when the ocean is depleted in calcium carbonate, and also can be very important in such settings because they'll be the main index fossils. And they're best known perhaps for forming radiolariite cherts. Conodonts, these are some more jaw apparatus for marine organisms which range from the Cambrian to the Triassic, but they're most common in the Ordovician and late Devonian. These are phosphatic in composition. So again, they're not preserved in palynological preparations. They get dissolved by the various acids that we have to put them through. And that's in contrast to those Scelechodonts that we saw before, which are preserved after we've gone through all the acids. So we have nanofossils as the last group, and these are tiny calcareous or siliceous microfossils. Most abundant types, but not the only ones, are coccolithophores, which produce the multiple elliptical calcite discs or scales, which you can see on that, that's the one, the top left. These are found in the fossil record from the late Triassic to the recent, and combined with foraminifera, nanofossils can provide an extremely high resolution biostratigraphic zonation, but being of calcareous makeup, both are not preserved below the carbonate compensation depth or CCD, so we have to be careful where we use them. So the next slide was the one we had before, which was this summary table of the amount of sample that you need for analysis. And as you can see, some fossils require much larger sample availability. Most importantly, those ones there are the conodonts. Unfortunately, in some parts, older parts of the section, conodonts are the most useful, but also need a huge amount of sediment. So we're currently sort of playing around, and Kath Gray, one of the major researchers in this area, are looking at using acritarchs in this older Cambrian to Devonian section where we don't have the spore pollen so we might be able to get away with using a lot less sediment. So you can see here that nanofossils and Palynology, we only need a very small amount of sample. And although I've got 20 to 50 grams there for Palynology, in some areas where we have really rich assemblages, we can get results from as little as 10 grams of sample. So, so in terms of samples, there are three main types of samples that we get this stuff from. We have core, sidewall core, and cuttings. Cuttings are probably the most readily available, but they are not as accurate, given that they're taken over usually between 3 to 10 metre intervals. And we also have issues with cavings there, which is where parts of the section already drilled falls down the hole and gets mixed in with what you're drilling at the time. So you have a little bit of uncertainty there. Sidewall cores give us a point sample, so that sort of eliminates the cavings issue, but they may not be suitable or representative lithology, because often sidewall cores are targeting sandstones for reservoir analysis, and sandstone is probably the worst thing you can collect for fossils. And then we have core, which is their best bit, because it gives us an excellent combination of a point sample and a full sedimentological data set, but that is expensive and rarely available. So in terms of, I've touched on before the best type of lithology. Grain size is the key here. The best lithology for collecting these fossils is a mudstone. So, and then at the other end of the scale is a very clean sandstone, which will generally be barren. So we tend to like mudstones, shales, even dirty sandstones in rich sections can yield fossils, but we generally try to avoid sandstones and carbonates as well. And also anything that's really quite oxidized, so red beds are also no good for preservation. Really the only thing that destroys these little organic fossils is oxidation. So we try and avoid that as well. So next slide there. So basically what we do is we get those samples, so sidewall core or cuttings, and we put them in a whole bunch of different acids, starting with hydrofluoric acid, hydrochloric, nitric, basically. I think they're the three main acids, and then we float off whatever's left, and whatever's left is mounted onto a microscope slide, and we have a look at that and note down what's in it. So this here is an image of the palynological range chart that is produced after we've analysed these samples, and you can see on the x-axis there are the different species. You don't need to be able to read it, but across the top there is the different species, and this is a particular group here of just the spores. And then on the y-axis is depth. So there'll be a couple of clicks here, which doesn't matter if we just click twice. I think we'll just put some lines on there so it's easier to see. So once the tax is plotted up, you can see the influxes, which are these, you can sort of see a couple of them there already highlighted. Gross assemblage changes, which you can see below that bottom one. You can see quite a big change in assemblage there. We also get extinction and evolution points. And it's these main things which form the biozones that we use to give an age to these samples. So I'll think the next couple of slides will actually go into biozones just to kind of tie it together. So that's pretty much what we palynologists do. And then we start to interpret the actual age of the rock, and that moves into the geological realm. So next slide there. So this is just a very quick background in biostratigraphy in Australia. Although palynology has been widely studied since the 1950s in Australia, it was in the 1970s and 80s that it really exploded onto the scene with the publication of the Helby, Morgan and Partridge Mesozoic zonation scheme. This produced a framework tied to absolute dating and global fossil events, which could be used across Australia. So that really changed the way palynology was used in Australia, because we were able to tie things together with a really robust framework. This is still largely in use today with a few modifications. Geoscience Australia published an updated Dinosis zonation scheme in 2004, which was again updated in 2006. And this was undertaken by the principal consultants, Helby, Morgan and Partridge, in collaboration with other biostratigraphers from Santos, Woodside and Geoscience Australia. So what I also wanted to note here on this coloured chart that sits in the middle there is we have the Helby, Morgan and Partridge original zonation, then we have a couple of other zonations there. And you can see this is the historical record, I guess, of the zonation schemes that have been used in the past. And they're generally pretty robust. It's pretty good, apart from in the Aptian there, where we've had a few changes. And this is really changes in the way people were classifying these zones. So it's just something to bear in mind when looking at historical palynological data, that you have to really pay attention to which zonation scheme was used. And something of a large amount of the work we've done in these databases that I'll be talking about next has been updating all that older data to a single palynological scheme. So what you're looking at compares to, like, you're comparing like with like, and not getting any confusion there with those zones. Okay, so these biozones that you just saw on that previous slide, these are directly related to the fossils themselves. So I won't go into detail on this, but I just wanted to explain that these biozones, they can be defined by the ranges of a taxa, or overlaps in them, or gaps in them, or acmes, which are just, I suppose, floods of a particular species. So in our palynological preparations, we get a lot of background fossils, which they may still have paleo-environmental significance, but they're very long range. And so they, kind of form the bulk of what you find in your palynological preparation. But we are actually looking for specific fossils which will give us a unique signal, and that's what defines the biozone. So out of those maybe 3,000 fossils that we find in our on our microscope slide, there may be only a handful of those that are useful as index fossils. So that's what we're searching for. So we kind of just sift through those and look for them. But you can also look at the gross assemblage to give you a bit of an indication of what age you're in. This is all tied back to the International Chronostratigraphic chart, which you'll all be familiar with. And this is done by various means, which includes radiometric dating and also ammonites in the Jurassic. So one of our jobs is to kind of keep this up to date, making sure that our biozones also relate to absolute time here. So there are GA do quite a bit of work with, and also UQ actually has done quite a bit of work with zircon dating to try and refine our ties between the biozones and the absolute ages here. So that's a background on palynology, just so you, trying to fit an overview of palynology into 40 minutes is quite rushed, I'm sure, but hopefully that gives you an idea of the fossils that we're using. And so now I'll move on to like how we actually use this data. So this map here shows some of the databases we've built over the years, and using a standardised paleozonation, we can relate all of these sections across Australia to each other by understanding the age of the sediments. So this data, forming these databases, is mostly all available from open file sources and data sets. We have included here things like borehole locations, basin and outcrop geology shaped files, wireline log data, biostratigraphy, lithostratigraphy, sequence stratigraphy, check shots, deviation, geochemistry, core points, core analysis, DSTs, RFTs, all that sort of stuff. It's all available from the government databases. It's just you need to find a way to bring it all in and put it all together so that it actually makes some sense. So if you're working on Australian projects, there are a number of data sources that you'll need to access. So if you're working in the offshore, the main one is NOPIMS, which is the National Offshore Petroleum Information Management System. And this is where all the petroleum drilling reports that are open file are stored. And then each state on the right-hand side there, each state has its own onshore database. So we've got here like the DIGS, people may be familiar with these already, the QDEX, which is the Queensland one, Strike is the Northern Territory one, WAPIMS is the West Australian one, GeoVic for Victoria, PEPS, South Australia, and the Mineral Resources one, I think that's called the Tiger database, I'm not sure, for Tasmania. So whilst these are really amazing, excellent resources, they're not always fully consistent with each other. So we generally need to do some work to kind of standardise these data sets, and as we bring them into the one single data set. So additional sources we always like to include, and these are not always available from the same databases, include Geoscience Australia data packs and studies. There's quite a lot of those on the Geoscience Australia website. There's also academic papers. A lot of these older academic papers are often hard to get, but they form a large part of our biostratigraphic databases because that's where a lot of the really detailed work has been published. We also have academic theses and geological survey publications. So there's a huge amount of data out there, and one of the biggest challenges is to bring that data into a form where we can actually understand it and use it. So that's the aim of our stratigraphic databases. We put all of this data into the context of time and age, so that we can start to build up a picture of the geology by age. So this is actually, to get to the fun part, we have a huge amount of work that goes into that. And this is something that takes the most time for us. And that is, we begin by loading up the location data and the Y-line logs. Government databases have fantastic location data, and most of it's updated to the latest geodatums. So, but there do tend to be occasional issues with duplications, particularly in areas where you've got petroleum wells, stratigraphic wells, mineral holes, and core CSG holes. A lot of the times, the whole names have been reused a number of times. So a lot of care needs to be taken to ensure that you're matching the right data to the right borehole. And that's something GA does a bit more of, because it gives you a universal well identifier, so that we can tie it all back, so we don't have wells matched to the wrong well. So the Y-line logs are often unmerged, and you can find them in all sorts of formats. So that's always been a big challenge. And I've got an example there in the bottom right-hand corner of the screen from the New South Wales MinView database. So they have some digitised logs there, and they're in a very unusual format that you can't readily load to most of your software. So, and they've also got, each well has a separate file. So a bit of time's taken, you have to load up those files and merge everything in. And then at this stage, we would also be loading up deviation surveys so that we can plot the wells correctly in space. So that final one in the bottom left corner is just an example of a pretty standard lithostrat table from a well completion report. So that sort of becomes our starting point for how we start to piece all these rocks together. But lithostratigraphic terminology has changed through time, with different companies adopt different formation names. So this is where we have a sort of a starting point, and then from here we want to try and get some sort of a standardised lithostratigraphic scheme together, so that when we look at these wells, we're comparing like with like. So, and at the top we have just one example of a Palmology report. So this is where it makes it very difficult for a non-palynologist picking up this data. Some of the later Palynology reports are great because they just give you a depth and an age, and that's all you need. And as long as you know what zonation scheme you're using, you can keep going. But occasionally you get, and particularly in the Central Australian basins, you'll get this type of report, which is very complex and just lists a lot of species. So you really need to know what you're looking at. So that's one of the things we've been working on in some of these publicly available databases, is turning that jargon there into an actual date with a depth next to it to make it easy to use. So my next couple of slides, they're going to show something of a case study using these now publicly available data sets. So the first one I want to talk about is this Otway Basin data set here. This has now been extended out to the offshore Otway, the Bass Basin and the Gippsland Basin, using the same stratigraphic framework. So as part of this work, we reviewed every single biostratigraphic report for the entire onshore Otway in the state of Victoria. And this is the first time a project of that size has been done on a basin-wide scale. So it was really quite exciting, but also quite daunting, because there's a huge amount of data to be extracted from the government databases there. So we aimed, then the secondary part of this project was to plug the data gaps across the basin we actually ended up taking a further 748 Palynology samples and 312 Micropal samples, predominantly in core that hadn't been sampled before. And then by combining these data sets, we're able to cover the entire section in the onshore Otway, using the Micropal predominantly in the carbonate-rich sections in the Cenozoic, and then the organic fossils in the siliciclastic sections. And so this was a really broad study to infill data gaps and to help with a big regional stratigraphy across the entire basin. So the results of this study, I've got the link on there. They're available from the GSV website. So there are data pack there which can be downloaded. So this next slide here, this is a really good figure which touches on what I talked about before about the various zonation schemes. So in this particular basin, we have 12 different previous versions of zonation schemes used at some point in time. You don't really need to see the detail there, really just to see that there are 12 of them. And this particular report here was a regional report done by one of the state governments, I think, which attempted to put together some sort of a chart that helps you to transform your data between the older zonation schemes into the most recent one. So there'll be a couple of clicks here. So if you want to just click maybe 2 or 3 times, and it was just showing how we have a zone, which has got, unfortunately, they reuse their zone names as well. So this makes it very difficult. So in this particular one, this D speciosus zone was in use from the 60s to the 80s, and then they changed the name of it, but they still used the actual marker that the zone was named after. So it made it very, very confusing. So we ended up with, you end up with a lot of names reused. And if you don't know what your original zonation scheme is, you can get very confused very quickly. So hopefully if you click again, it's going to take us to the next slide, which makes a little bit more sense. Okay, so here we go. So this is an example of some of the issues we found and how we sort of resolved those in this new data set. So what I've got here is 3 schematic wells, A to C, with shale and sandstone interbeds. And the question is, how do they correlate? So at first glance, if you just plot the biostrat direct from the reports, it looks like there's no correlation at all. And so this has been one of the main problems with Palynology falling out of fashion somewhat in some of these basins, because you look at that and it's not helping you. However, if you look at the zonation scheme used in each of the wells, you'll see that they're all very different. So we need to standardise the results. So the next click should show you what a standardised result is. So after we review this data, standardise it to the same zonation scheme, and in this case, we've standardised everything to Morgan 2002 zonation scheme. And now you can see that like is comparing with like, and the boundary between the 2 zones sits somewhere between that sand layer in the middle there, indicating that the sand is likely correlates across between those wells. So you can sort of see from that very simple example that by standardising the legacy data to the one scheme, you can have a really big impact on how you use the results. So getting back to this stratigraphic framework, this is really what ties everything together. So yeah, I'll tell you when to click for these because there's a few to click through on this one. So so quite early on in the building of a database is really like to establish some sort of at least a basic stratigraphic framework to work within. And that does evolve over the course of building the database, but I really like to get something in place early on. So a good place to start are the charts published by Geoscience Australia. And this example looks like an old one because it still has AGSO written on the bottom. But this is an example from the Otway Basin. And you can see on the left-hand side, you have that coloured section is the timescale. And then all those yellow sections are the various zonation schemes. And that's different microfossils as well as different vintages of zonation schemes. So there's a whole lot of information there. And then on the from the centre to the right, you've got all the lithostrat schemes. So this can be a little overwhelming at first sight. So I always like to try and simplify this down, because if you've got too much lithostratigraphic nomenclature, again, it's very difficult to move regionally across the basins and from basin to basin. So if we click on the next one, I always like to go through all the reports and collect up everybody's zonation schemes and stratigraphic columns. So here's a nice example from Origin Energy from a well completion report, but this is quite specific to the shipwreck trough in the Otway Basin. But we do start to see here some of the issues with the lithostratigraphy in a time sense. So if you have a look down, probably about, pretty close to where your mouse is right now, the Skull Creek Parati formation there, you start to see how some of the lithostratigraphy is time transgressive. And that in the past has caused problems with correlating sands to sands, because you can't really, it is very difficult to do it lithostratigraphically. So that's why we use the biostratigraphy. So it can give us an age to those sands, and then it helps us to tie those wells together. So next click. Should bring up. Yes, this one. This is one of my favourites because I love sequence stratigraphy, but a lot of, I'm not sure how much it's used in the minerals world, but we love it in the petroleum world. So this is where this comes in. Again, this is another Origin Energy chart from a well completion report, but it displays a lithostrat in a sequence stratigraphic framework. So that coloured curve there is the main thing of interest, which is a coastal onlap curve, which is just showing you a sea level going up and down, pretty much. And you have sequence boundaries, which are those little wiggly unconformity lines there. So what they're basically trying to do is to pull the lithostratigraphy into a time framework, and that helps us to tie from well to well. But the main thing we use to do this is, again, biostratigraphy, because that gives us an age for our unconformity surfaces there. So one more click gives me my favourite chart, and this one comes from Woodside. Woodside have historically done some of the best work in the sequence stratigraphic world, and some of their strat charts are some of the best ones I've seen. So this one just shows part of a giant strat chart that goes across into the Gippsland Basin. But again, you can see on the, it's quite complex, because on the left we have the time scale, and then we have various zonation schemes again. We have the HARC curve, sea level, global sea level curve, and then we have the lithostratigraphy in there. And so what they're trying to do there is put that into a sequence stratigraphic perspective. So you can see those lines going across, the green and the red lines. That's the sequence boundaries, transgressive surfaces and maximum flooding surfaces. So that's what we're trying to do using the biostratigraphy, is kind of define those time surfaces, so you can slice through your rocks in a time sense. You've already got one of these, and this is a really good one. In the Otway Basin, we've got this where other people have done work, but then other places we don't. So then we have to start building this as we're building the database. So that's why it's important to get at least something basic in place as you start to load the data. And once you have that broad framework in place, if we click once more, so this is a very basic one, where we're starting to standardise things and just make it a little simpler to understand. So we're taking out a lot of those little tiny sand names often, and especially in a turbidite environment, people will give each turbidite lobe its own name, and it gets really confusing. So what we've done here is just got to take out that variation and just kind of mould things into a chronostratigraphic sense. And you can see in where should be able to see, there's some lines going across that lithostrat thing there. In the blue section, there's a couple of lines in there, and they're little the bottommost blue one, there's some little lines. Those lines there, yeah, there's little lines going across there. These are actually biostratigraphic flooding surfaces, or what we interpreted as flooding surfaces. So in the actual palynological preparations, we get almost a bloom of a specific bug. And if we know enough about that part of the section and those bugs, sometimes these things can be indicative of a major change in environment. Usually it's a flooding surface. So this one here, these are both candidates for maximum floods. There's a couple of ones at the bottom of that next brown block. Those ones, that one is a candidate for maximum flood, and that's also somewhere around there is the PETM, which I've forgotten the name of, I've gone blank. So we have some candidates for maximum floods, and that gives us a way of correlating from area to area and around Australia as well, because we can tie them back to the global sea level charts in many cases, although sometimes they obviously are tectonically influenced as well, but they provide excellent tie points. So once you have that basic framework in place, then we can start looking at the Pali in more detail and getting more information out that can increase our resolution. So the next slide will give the bit that I like the most, which is the well-to-well correlations. So these two steps are being done almost concurrently. So we have a first pass stratigraphy, and then we're going to go into the well by well details. So what we've got here plotted up is all that open file data plus our interpretation. So we have the wireline logs, and then in the far left column on each well there, that's your biostratigraphy. So in black we have, far right, we have the Palynology in black, and in blue is the MicroPal. And then on the far left, we have interpreted and standardized lithostratigraphy. And then we have a couple of little columns in the middle there where we've got two different versions of a sequence stratigraphic framework. One is the Woodside one, which is quite popularly used in the Northwest Shelf, and the other is the Santos one, which is the main one used in the Otway Basin. So what we can do there is we can start to correlate across a lithostratigraphy in a time framework. So the next slide will show that in a little bit more detail. And all of this is also available for download. So here we have where we start to have a little bit of fun and get excited with the Pali. So what we've done here is we have the main lithostratigraphy and chronostratigraphy, which are the coloured columns on the left of the gamma ray log there, and then these various grey sections is where we've gone into the Palynology itself and pulled out individual events which are inside the biozones. So they're higher resolution than the actual biozones, and then we can start to correlate individual sands based on these assemblages and whatever bugs are in them. So it can get really detailed. Once you have that regional framework, then you can start focusing in on areas of interest. And this particular one here is looking at some sands within the Belfast Formation in the Otway Basin there. So I'll go to the next slide, and I think this will be the second one. So that was a largely marine section. We'll talk about a largely non-marine section here. So this is an ongoing project that I'm working with at the moment, part of a really interesting project being undertaken by Geoscience Australia. The learnings from the larger, well, what my part of this is a very tiny part of this larger study, but the larger study itself will be presented at the upcoming CABS symposium. So I'm looking forward to seeing that because I've only seen the biostrat side. This is a big hydrogeology study. The reports that we have so far are available from the Geoscience Australia website, which I've got linked there. So I've just summarised here some of the data sources. So one of the biggest challenges for this study was the absolutely huge amount of data available. I've just listed some of the main ones. These are the state government databases. We also had a huge amount of data coming out of Geoscience Australia from their own databases, the StratDat database and all their other databases that they also have available. Again, all of this open file data. So as part of our study, we also included academic theses, geological survey reports, and a whole load of published papers. So these are often not available inside these state government databases. So we really do have to extend our search outside of that into everything that's available. So this region is very heavily explored, and there are around about 80,000 wells across the Great Artesian Basin, and that includes petroleum wells, CSG, stratigraphic, minerals, and we have data from the 1960s right through to present day. So a lot of data. So the next slide, I just want to have a little, show you a little bit of what is available, just because it's quite exciting. This is the South Australian Sarik database, and I want to show this one because this is one of the few databases where we've got a palynologist looking after the data. So it's quite exciting, and it makes our life a whole lot easier when we want to look at biostratigraphy. So what we have here is you can search the Sarik database for a well. So if you click once, we've found the well skeleton 2 there, and it has a biostrat data set available. So if you click on that, that then gives you a list of samples taken from that particular well and who collected them. And then if you click on that again, you click on those results, that'll take you to a summary of the rock that was taken. And then if you were to click again, there's a biostratigraphy tab, which in this particular sample wasn't filled out, but that gives you all the details about that particular sample. And in some cases where they have the actual species, they'll have them listed as well on that page. So it's a fantastic resource, but one of the biggest problems, and it is often the problem with big databases like this, is what do you do with that data? Like, you've got to find it first, and then once you've found it, what do you do with it? So that has always been the biggest problem with dealing with the data in the central basins is, finding a way that we can understand it and use it, because it's just the sheer volume of it is quite daunting. So if we click again, this will get us back to what I was talking about with the Artesian Basin. So this map here is just a summary from the first phase of work we did in the data gathering phase and building the initial database just to get you started. So we have here a shapefile for the Great Artesian Basin, which is that green outline. And in the colours, we have the sedimentary basins which lay underneath that. The green dots are the wells for which we found some sort of palynology, and the red dots are wells we looked at or included in the study which had no palynology. So you can see here there's some pretty large data gaps, and so the later phase, which we're currently working on now, are aiming to fill that in. So eventually what we hope to have is every single palynological study that's ever been done in the Great Artesian Basin polygon will be included in a data set where you can just download the Excel spreadsheet and load that up to your database. And at the same time as this, my colleague Katie Norton, whose report I also linked there, she is doing the same thing with the lithostratigraphy and the wireline logs, so that eventually you'll be able to download all this data in one nice clean set and just load it up to whatever interpretation software you've got and use it. So as with that previous study that I talked about, we started loading the raw data, reviewing the Palynology and gathering information on the stratigraphy. One of the main problems we had with this particular basin is it's been heavily explored by Santos for many, many years. And so a lot of the information is not available outside of Santos. So we're trying to piece together a lot of information, really starting from scratch, because there's not a lot out there. So while we were building this database, we're also trying to build the framework at the same time. So this here is another Geoscience Australia chart, which was again the starting point. And again, we have the same format with the timescale and then the various zonation schemes. And again, what we tried to do is simplify that biozone column down. So we just got the one or two columns just to make it simple. And we reviewed the Pally. So we've just got that one, so we can, compare like with like. Whereas if we were to retain the old zonation names, we may have 5 or 6 different zonations that you're trying to juggle between. And then we have, of course, the lithostrat on the other side there. So if we click another time, we should get another chart up. So then I've got some more. Again, we went through all those well completion reports and collated up all of the various stratigraphic charts that different companies and different researchers have made. And here's an example here of one from the Sarik Basin from a well completion report. If we click again, we'll get another one. This is one of my favorites. I really, really like these older ones. They give us a lot more information, and they're where we can find out where these odd localized nomenclatures come from, because often we'll find some of the old reports have got names that are no longer in use. So these are really useful for us to kind of translate the names. But the only problem with these, often they don't have any biostratigraphy. So we've got to work out where they sit in the section. So although they look good, they can be quite confusing. And then if we click again, we might get another one, hopefully. Yeah, and then we've got these really detailed ones. This is just the Bowen Basin, so it was below our area of interest, but we collected this up as well because often the Palynology goes across the boundary into the basin below it. So we were collecting, as part of this, we didn't just stick to the Jurassic Cretaceous Palynology. If a well went right into the Permian, then we reinterpreted the Permian as well. So there is actually some, older data sets available in the one that we can download today. So next click. So here is really what our first pass understanding of what is going on. So what we tried to do here is to capture the main formation names used, also while trying to fit these into a chronostratigraphic framework. So this chart is largely based on charts that we have seen in the open file arena from Santos stratigraphy charts, but not all of them are available publicly, as well as incorporating what various researchers say. For example, some of the work that Neville Alley and Steve Hoare in South Australia were doing on the southern part of the Eromanga Basin there. Also, a lot of the work that John McKellar and also the University of Queensland were doing in the Sarik Basin. So we tried to tie all that in. There's various researchers doing zircon dating. For example, I'm not sure if it was James Cook University, were doing some work on zircon dating in the Carpentaria Basin. So we tried to incorporate all of that to try and pull all these zones into one single framework. So that was the first pass there. And then down on the right-hand columns there, you'll see the chronostratigraphy, which is all the sequence boundaries, I guess, that Santos were using. And that's because it's the most commonly used. They did the large amount of data in there. So that's why we used that one. And then we have on that far right column, that is the hydrogeological units. And I won't go into detail on that because that will be discussed more in the upcoming CABS presentation. But the main reason for us doing this work was because they needed a consistent framework for tying their hydrogeological data across from the Carpentaria to the Sarak to the Eromanga Basin. So that's the main reason we were starting to look at this and have a look at the Palynology to try and allow us to tie across these basins. So this next figure, this comes from Katie Norton's report, which is the other report that I linked on there. Oh, sorry, no, this one doesn't. This is actually mine. The next one after that does. So this figure here is just where we tried to identify wells with the most and the best quality biostratigraphic data to use as key wells, which help us to refine the biostratigraphic framework. So we highlighted a lot of these wells and reviewed this data, and some of it was infilled with extra samples to try and really get a good framework in a biostratigraphical sense. The next slide should hopefully show what are the figures from Katie's report. Yes, here we go, which is touching again on what I was talking about the different lithostrat in the different basins. So you have the far western part of the, above the Paderca Basin, I guess, which is the Algebukton sandstone, which is a whole pile of sand. And then as you move across into the Cooper-Eromanga Basin, you have different formations appearing, Merton, the Mer, which most people who've worked Cooper and Eromanga be familiar with. And then as you go across into the Sarak Basin, you have your Arello and Gubbermundas and all that sort of thing. So it makes it very difficult when you're trying to kind of make these layers, to build these layers with a different lithostratigraphy. So Katie's job here was to get that lithostratigraphy right, because that forms the basis of most of our correlation. And then we use the biostratigraphy on the next slide, hopefully, to give us some timelines. So those red lines through there, these are the chronostrap. This is the sequence boundaries, I guess, as we call them, that Santos are using. And so we use the biostratigraphy here to sort of cut through these lithostratigraphic units. And in a way, we also adjusted the lithostratigraphic boundaries so they tie a little bit more to time, so that you can start to get some time layers in your sections. Okay, so these are ones we can download, and this is just absolutely amazing work. I've never seen anything quite so detailed. And this is some of the work that Katie has been doing. And she's made these massive transects that really run through the Surat, Galilee, across the Cooper to the Paderca. So this is just looking at above the Triassic. So it's the Mesozoic section. But she's correlated from well to well. We use the Pali here and everything else that we can find, and created these massive sections correlating all these wells. And then, and again, this is a data set that can be downloaded. So all these formation tops here can be downloaded to load into your own interpretation software. And it really is a lovely data set. So this is kind of what is still ongoing. So our next slide, I just wanted to show some more ongoing work. Okay, yeah. So this one came about as part of a side project, as part of this whole Great Artesian Basin study. And this was looking at the Lake Eyre Basin, which sits above the Great Artesian Basin. So one of the questions we sort of had is, can we correlate those sections as well? And again, this is a larger project that I wasn't involved in, but my input was this, just to input the Palynology. And so one of the questions asked is there anything that can help us to identify which parts, of the section we're looking at in the in the Lake Eyre Basin? So our first step, of course, was to collate all the Pali data and go through all the reports. And we put together a sort of a framework here with our stratigraphic units, our biostratigraphy, and then the time scale. So the second click should give us a map, hopefully. Yes. So what we've got here is the pale yellow is the Great Artesian Basin, and then that darker yellow on top is the Lake Eyre Basin polygon. So what we were able to do, because we had this stratigraphic database, is we could query our existing database and tell it to find us all wells which have Palynology in the Cenozoic. So that's what we did, and we came up with, there's not a great deal of data outside of the Frome embayment there in the south part of South Australia. It's not a huge amount of data, but what we could find was quite interesting. And what we also found, and this kind of alerted us, I guess, to some of the gaps in our data. In our previous database, we concentrated mainly on the petroleum wells. But once we queried this database, we started coming up with some Pali data that had come out of the Sarik database that was alerting us to a lot of the mineral holes in the Frome embayment for the uranium drilling contained a lot of Palynology. So we were able to go back to the Sarik database and start looking at the minerals data sets, and that's a lot more complex because it's not as easy to find things. We have to basically go through report by report. And we did this as well for New South Wales, Northern Territory and Queensland, looking for anything that had Palynology in it. So that was a use of the stratigraphic database. And then we also started adding in minerals and water boreholes as well. So there's quite a lot of data out there, but once you have a framework, it's quite easy to query it for more detailed data. So if we click one more, I think that's the end of it. No, that was the, okay, so that just shows us there. That was one of the, this is another report that'll be coming out fairly soon. And this just shows where once you plot the Pally on there, you can start to see that underneath that, you've got a yellow and an orange section that goes all the way across. That's the Lake Eyre Basin. Underneath that was our existing database showing all the Cretaceous and Jurassic formations. So yeah, that was our starting point, and then we were able to pull out more data to fill in the gaps in the Cenozoic. So that showed us that having that framework made it much, much easier to move to a different area. And that's what we're hoping will happen if we have to move into things like we've already started doing that with the Paderca Basin. So the Paderca Basin, I think that's also going to be presented at CABS, a larger study. But again, this was, what have we got in the Paderca Basin? We were able to query this database and find all the data that's readily available. So the last click, I think, is the last slide. And really hoping to bring this around to these little bugs and how tiny they actually are and insignificant seeming that they are. They can lead us from little bugs in little bits of dirt to the next click, which is the big correlation. So hopefully that gives you an overview of biostratigraphy and exploration and how that can be used. And hopefully if you ever need to use it, there's plenty of free stuff out there to be found. So thank you for your time. And hopefully if you've got any questions, please let me know.
Thank you so much. That was so interesting and I've learned a heap. So I really appreciate that.